And so my task in this session is to first talk about the future of proteasome inhibitors, and then Dr. Gertz will come and talk about how he treats myeloma today, and then I'll return to talk about immune therapy in multiple myeloma. So, so the future of proteasome inhibitors in multiple myeloma. So we have got now three approved proteasome inhibitors in multiple myeloma. Bortezomib, which everybody I think is familiar with, has been around for the longest period of time. Carfilzomib thereafter, and ixazomib. The other three shown here, the one on the third row, the cephalon compound will probably not be pursued. I think it had rashes, and the company chose not to manage the toxicity and just stop development. But there are two others that are in development, oprozomib. That too first was used a few years ago and led to a GI toxicity profile that was deemed unacceptable, but now is being resurrected with a new formulation, which seems, again, we haven't seen any data, but it's probably a little bit better, certainly. And then we have bortezomib, which continues its path towards development. It was introduced into clinical development much at the same time as carfilzomib, has got CNS penetration, and there are some moves to try to develop it as a CNS penetrating drug. So, but my talk is going to mainly be focused on bortezomib, carfilzomib, and ixazomib. And as everybody, I think, is aware, the paradigm of treatment in multiple myeloma over the years has certainly evolved. It used to be that we used to give treatment to best response and then give a treatment-free interval. But I think that most people in the field now are in agreement that this is a disease where continuous therapy does seem to pay dividends, whether it be in the transplant-eligible paradigm or in the transplant-ineligible paradigm. Though not conclusively proven by prospective clinical trials, meta-analyses have suggested of trials that give this as a continuous therapy or as therapy interrupted by periods of being off drug and then restarting, that those that got continuous therapy on clinical trials did have longer PFS, PFS2, and overall survival. And this was the subject of a publication by Palumbo in the JCO a few years ago. So, how does proteasome inhibition fit into this whole paradigm? It has a role in literally every aspect of this. So, the way I'm going to talk about things is first talk about the frontline treatment and the role of proteasome inhibitors in transplant eligible, ineligible, and then go to the relapse refractory disease. Focus more on the recent trials that have come out and summarize some of the older data. So, bortezomib-based induction pre-transplant is the standard of care. This slide just summarizes a series of trials that showed that proteasome inhibition, compared to old standard of VAD chemotherapy for most part and some thalidomide-based regimens, did lead to improved progression-free survival in every case. Overall survival data at the time of publication of these studies was not mature enough to show survival advantage. The data that is more recent with proteasome inhibitors in the frontline setting is trying to answer the question of whether transplantation is still a necessary part of upfront treatment. And I think this is something that was reinforced to us by the IFM study that looked at RVD given as continuous treatment versus giving RVD for 4 cycles pre-transplant, then giving a transplant and consolidation post-transplant, that there was improved depth of response in those who got a transplant, but also improved progression-free survival, as can be seen here. So this hazard ratio of 0.70 led to the DSMB unblinding the study and allowing patients to cross over, if need be, to the transplantation arm. So I think that in this era, transplant still remains part of the paradigm of treatment with myeloma. We certainly have made major progress in the treatment, and every time a new drug comes out, there is always a question raised, is transplant still necessary? And I think that the best results still are when we sequence our drugs in the transplant paradigm. So it should be a transplant and new therapy paradigm rather than transplant or new therapy paradigm. Now, what about in the post-transplant setting? What do we know about proteasome inhibitors in the maintenance phase? And though the standard of care for maintenance therapy is lenalidomide based on more randomized clinical trial data, there is data looking at proteasome inhibitors. Bortezomib has been looked at in multiple randomized clinical trials. The data from the Sonneville study, shown in the first row there, showed that there was an improvement in progression-free survival and overall survival for bortezomib when given pre-transplant and then repeated post-transplant compared to a regimen which had no bortezomib in the pre and post-transplant therapy phase. The thalidomide was the maintenance in that study, and that study showed that there was an improvement in outcome for patients who got bortezomib. But the fact is that we do not know if the benefit of bortezomib came mainly from the post-transplant phase of maintenance or whether the pre-transplant phase also helped. So it's not as clean a study, and more recent updates of this data seem to suggest that it is those that have high-risk disease and get a tandem transplant and go on to bortezomib maintenance may actually be the ones who benefit the most. The other couple of trials, again shown here, did show some benefit to the use of bortezomib as maintenance therapy. In the real world, most institutions will these days combine bortezomib with lenalidomide for high-risk patients. There is no randomized data to support this use, but the fact is that a trial done at Emory University looked at giving RVD. They even gave the DEX for 3 years post-transplant, and as you can see, the curves in the high-risk population do look pretty impressive. This was only high-risk patients. So the curve number D is those who had 17p deletion. That, as you know, is one of the poor prognostic factors. So 414, 1416, 1420, deletion 17p is something that most institutions try to give dual maintenance therapy for. Though recently data is emerging that the only people who have got adverse prognosis due to 17p deletion or 414 translocation are those that have either a mutation of p53 on the other allele or a mutation in FGFR in the case of 414 translocations. The issue is that that is not yet clinically a test that we can easily get, so we have to lump all the 414s and the deletion 17p's into a somewhat poorer prognosis category. What about carfilzomib? So we've talked about bortezomib in the pre-transplant setting and the post-transplant setting. Carfilzomib has been looked at now in more than one study. Most of the work has come through Dr. Jacobowiez's efforts, led through the Multiple Myeloma Research Consortium at the University of Chicago. He has done 2 serial studies, one wherein kyprolis, Revlimid, and dexamethasone was given, and then patients underwent a transplant, which is on the right-hand side, on your left-hand side. And the other study was one where only KRD was given without transplant for extended periods. Those who were enrolled on the transplant study also got consolidation with KRD and maintenance out to 18 months. So it was a pretty intensive treatment regimen. And what these are obviously not randomized studies. These are 2 trials that were done in succession, and curves have been superimposed to show that those who got a transplant seem to do better in terms of PFS and OS. But again, small numbers of patients, we can't make too much out of this data. But median follow-up was shorter in the patients who got a transplant, 2 years versus about 4 years. What has come out of this as a matter of a lot of conversation is the high rates of MRD negativity, and that is something that most trials are now starting to look at. We look at it either by multicolor flow cytometry or by next-gen sequencing. And the rates shown here at the end of 18 cycles by NGS of 74% MRD negativity are pretty impressive. But as you can see, even after 8 cycles, 64% had got there. So the question is, do you need to go 18 cycles in everybody? The issue of MRD negativity as a goal for therapy, I think, is very contentious right now. Certainly in clinical trials, it is being explored and needs to be explored. Outside of the context of the trials, I think the field is pretty split. There are those that have started using it in patients and actually even making treatment decisions to intensify treatment in those who are MRD positive to get them to MRD negativity. But that at the moment isn't supported by any evidence, and I personally don't do so. So, what about trying to further improve upon the transplant paradigm with proteasome inhibitors? So, daratumumab uh, obviously is a drug that just got approved for the non-transplant eligible patients in frontline setting, and we'll review that data in a minute. But over here we see again from Dr. Jakubowiak's group a study looking at adding KRD and data together, and very short follow-up. Can't make too much out of this data presented at ASCO last year, but again, a pretty impressive overall response rate with 91% VGPR and 43% CR rates. Whether this is better than KRD alone is a little bit difficult to say, plus the follow-up is short and these depths are likely to get deeper. There are certainly other trials ongoing in the transplant eligible population trying to further define the role of proteasome inhibitors and monoclonal antibodies. And I just put up the two larger studies that are currently accrued. One is a study looking at RVD plus minus DARA, followed by transplant, followed by randomization again to RVD or RVD DARA. We've just seen very preliminary data from the RVD DARA combination in phase 1-2, but haven't obviously seen anything in terms of efficacy. And then there's an ECOG study trying to see if KRD is really better than RVD. The field there too, I think, is somewhat split. There are those that have started using KRD. It is in the compendia at a level that most Medicare patients and insurances do get reimbursed for. So in my own practice, I have started using KRD in frontline patients if possible. I think that there is some reluctance in some quarters because of increased risk for thromboembolic events, but the fact is that at least in my own practice, I haven't seen any sense that that is higher than RVD, and certainly Velkid's neuropathy is certainly something that one wants to avoid as far as possible. So if not for proven efficacy, even for, I think, tolerability, KRD to me makes more sense. What about the role of ixazomib in the transplant paradigm? This is an oral proteasome inhibitor, so seems best suited for patients who have need for long-term treatment with maintenance. And MD Anderson did a study looking at ixazomib with lenalidomide in the post-transplant maintenance phase, small study, essentially looking at the feasibility of this. This was supposed to be the basis for a larger phase 3 study of ixazomib, lenalidomide versus lenalidomide, which is the standard of care. But that was shelved at the last minute a couple of years ago. It was supposed to be an 1,800 patient study, and nobody had the appetite for it. And with the advent of daratumumab and monoclonal antibodies, people thought that those were probably the better drug class to pursue in large randomized studies, and that is where the field has moved to. In our own center, we are leading through the Multiple Myeloma Research Consortium, 10 institution effort to compare ixazomib to lenalidomide. This is a study where patients who get a stem cell transplant actually get consolidation with ixazomib Rev-Dex for 4 cycles, and then get maintenance with either ixazomib or lenalidomide. It's a 200 plus patient randomized study, and obviously it will require a little more time to know how this experience pans out. What about for patients who are not transplant candidates? For them, we know that with the VISTA trial, VMP versus MP, there was certainly an improvement in progression-free and overall survival. and that became a standard of care for patients in Europe. In the United States, never did take off much because of the reluctance to give an MP-based regimen and the ability to give alternative regimens like Revlimid and dexamethasone that has been possible here for a long time. So the fact is that in patients who are not transplant eligible, certainly trying to give non-melphalan based regimens is now something that has even taken off in Europe. And what we see is then how do we improve upon an RD standard? And there, this study done by the SWOG was looking at a population of patients that were not headed to transplant. Some of them may have been transplant eligible actually, but this study showed that VRD compared to RD did have a progression-free survival and overall survival advantage. For the longest time, we used VRD without much data to show conclusively that it was better than RD, and there was always skepticism that we wouldn't be able to show survival advantage in patients with myeloma in studies, but this study, and as you'll see some other studies later, have broken that kind of fear. So what we have here is VRD versus RD. But in the patients who are truly not transplant eligible, I think there are two groups, those that are between 70 and 80 and those who are above 80. Above 80, I think most people would still stick with currently a two-drug regimen of Revdex, but between 70 and 80, the patients are often strong enough to get a three-drug regimen, and so the regimen of VRD light, wherein you dose attenuate the Velcade, Revlimid, and dexamethasone, is probably the most popular regimen, though it is just a small study of 50 odd patients that we have in the literature to go by. I think for that population, VRD light at the moment is a regimen that we use very frequently. What else has happened recently in the non-transplant eligible space is the approval recently based on this study of daratumumab. So daratumumab VMP versus VMP, and this study showed that patients had improved progression-free and overall survival with a hazard ratio of 0.50 when daratumumab was added to VMP. The issue, once again, is that people do not want to use a melphalan-based regimen, and whether this will have much traction is an open question. Some are hoping that one can maybe drop the melphalan and give dara with Velcade and a steroid. That would be more palatable, and that could even be a regimen that would be possible to use in a transplant eligible population. But the thing is that this regimen did show not only improved PFS but also depth of response. Um, the study that is awaited, looking at frontline non-transplant eligible population, is a study of ixazomib, Revdex versus Revdex, which is expected to read out within a year. But there are two other studies also in this population that will read out around the same time. One looking at Dara Revdex versus Revdex, and another looking at Elo Revdex versus Revdex. So we will have, I think, a dilemma which we'll need to sort out if we have all three approved around the same time as to what may be the best regimen. In the relapse refractory space, we have had a number of clinical trials recently read out, and I won't go into much detail about these. These have followed two broad paradigms: to improve upon a RD standard and or to improve upon, as shown here, a bortezomib dex standard. And these studies looked at Kyprolis dex versus Velcade dex, Daravel dex versus Velcade dex, and there was panobinostat versus Velcade dex versus Velcade dex that all led to the approval of the arms that were studied. And again, how we treat relapse refractory disease may be best left for the panel discussion because, and I think Dr. Gertz may also try to tell us how he uses all these different regimens that we have in the relapse refractory space. trying to improve upon the lenalidomide standard using proteasome inhibitors also has led to a number of trials, KRD versus RD, IXA-RD versus RD, and then we have other trials, but we're focused more on the use of proteasome inhibitors, so I highlighted the first two rows. And this, again, is something that is hopefully something that Dr. Gertz will discuss as to how he chooses based on all this data that we have in myeloma. Pomalidomide combinations with proteasome inhibitors are also popular. Carfilzomib, pomalidomide, dex is a commonly used regimen. Ixazomib, pom, dex is a convenient oral regimen. In this regard, just winding up, the latest data on the use of proteasome inhibitors in a large randomized study to be presented was the Optimism study at the ASCO meeting earlier this month. This was looking at a regimen of pomalidomide with Velcade dex compared to Velcade dex. And here we see, as expected, that there was an improvement in progression-free survival, the primary endpoint, with pomalidomide Velcade dex compared to Velcade dex, giving us yet another option to treat patients with. Um, and then the other study at the ASCO meeting that got presented for the first time was a randomized phase 2 SWOG study looking at carfilzomib at 27 milligram per meter square, comparing it to 56 milligram per meter square given on the conventional twice weekly dosing. And this study, the conclusion was there was greater depth of response when you gave the 56 milligram per meter square, but there wasn't any improvement in PFS or OS. There were more AEs, not the cardiac AEs, that were higher when you gave 56 milligrams per meter square of carfilzomib. The other study that was presented at the ASCO meeting was a study looking at once weekly carfilzomib dosing, and here the dose employed in the arm being studied was 70 milligram per meter square. As you know, there was a phase 1-2 study published in Blood about a year ago now that showed that 70 was the MTD when given as a single agent, and so carfilzomib dex were at 70 versus the conventional twice weekly dosing of carfilzomib at 27. There was higher dose delivery when you gave the once weekly at the higher dose, and there was an improvement in PFS, 11.2 months versus 7.6 months, with a hazard ratio of 0.69. So I think that this is a dose that isn't yet FDA approved. We need to see the full publication because there's been some concern about higher doses of carfilzomib producing more side effects. The depth of response was better, but at least in this study, the adverse events were no different when the higher dose of carfilzomib was employed. So to conclude, proteasome inhibitors are currently the backbone of frontline therapy for both transplant-eligible and ineligible patients. Maintenance therapy data with bortezomib for high-risk disease is encouraging. High MRD negative rates with extended duration carfilzomib have been shown. Ixazomib offers a convenient oral option, and for relapse refractory disease, there's a multitude of treatment options, and we need to somehow personalize it for patients, and that is hopefully something that Dr. Gertz will tell us about. So without taking any much more time, I'll go ahead and introduce Dr. Gertz. I think he